Hi guys, so we're going to talk about blood today, and this is going to get in our section about the cardiovascular system. Now, blood is a life-sustaining transport vesicle. It is considered a connective tissue, and it's important because if you go into a clinical setting where you're dealing with patients, the physiology and the anatomy of blood will help you to advise patients on activities to better prevent things like blood clotting during hospital stays. So in order for us to truly understand how the body functions, we kind of need to understand blood a little bit. Now, blood has three major functions. It functions in transporting, regulation, and protection. And each one of these is going to have a few parts to it. So the very first thing we want to look at is transport. And when we're talking about transporting, basically what is the main thing that we think about that is dissolved into the blood that moves around the body? Hopefully you realize that I'm talking about oxygen. So it helps to deliver oxygen, but not just oxygen, things like nutrients to body cells. Things essentially carbohydrates or proteins or anything that the body or body cells may need to build molecules or to allow for the production of energy. Now, if it takes important nutrients and oxygen to the body cells, then it must also take things away from the body cells. So it's important in transporting metabolic waste. And they move those to the lungs and the kidneys to help them to eliminate them. So we're talking about things like carbon dioxide or even uric acid or urea. Now, we just got done talking about the endocrine system, so these guys are also very important in transporting hormones. And they go from the endocrine system to target organs. Now, on top of transporting, like I said, it's important in regulation. So, moving blood around is important in helping to maintain body temperature. And they do this by absorbing heat from certain parts of the body, right? And then distributing that heat outwards. It's also important within the blood. They're going to have things like bicarbonates. And if you remember from last semester, or if you haven't, that's fine, that those things are important in buffering the pH. So blood has the capability of keeping pH levels normal using buffers. And basically those are chemical buffers, like bicarbonate. And the last way that essentially it helps with regulation is throughout it, if we remembered, we're mostly made up of water, and a big amount of our blood is composed of plasma, and plasma is mostly water. So this is important in maintaining adequate fluid levels, essentially in the cardiovascular system. And I'm just going to abbreviate that by CVS. So in here we've talked about transport and we've talked about regulation. What's the last major function? Hopefully you remembered it was protection. So the protection functions that we have include preventing blood loss, so within the blood itself, it has things like plasma proteins and platelets, and in the blood. And these things help to initiate clot formation. So that essentially helps us to prevent blood loss. The blood is also important in preventing infection. And we have several agents of immunity that's carried in the blood. And a big part of our blood incorporates what are known as white blood cells, or leukocytes. So some of the leukocytes are important in creating antibodies. So you have antibodies that are there to help recognize anything that's foreign. We're going to talk later, but we have complement proteins that are there to help with protection. And then, of course, the white blood cells or leukocytes that we're going to get to. So the main three functions, transport, regulation, and protection, are all the main three functions of the blood. And they're there to help get important components that our body cells need to get rid of trash and waste, to help make sure that we have enough fluid in the circulatory system, to help with transportation of things like hormones, and to help protect us. Now, composition of blood. Blood is the only fluid tissue in the body, so blood's considered an actual tissue. It's a connective tissue, if you remember from the histology section in part 1 of this course. The matrix part is non-living fluid, and this is called plasma. A big part of the plasma is essentially water. And basically, plasma incorporates about 55% of the blood volume. Now, within the plasma, there's living cells. These living cells are what are known as formed elements. Right? You can see those. And basically, they're just kind of suspended or floating in this plasma. And there's 3 types. There's erythrocytes, which are red blood cells. There are leukocytes, which are white blood cells. And there are platelets. Now, platelets aren't complete cells. They're fragments of cells. And so with that being said, let's actually look at the 3 main components here so that we can see. Now, how have they decided what are essentially the blood and how it's made up? Well, they can extract blood or withdraw it and place it in a tube, as you can see here. And then they're going to centrifuge that. And what that is putting these tubes in a piece of machinery that spins them around very, very quickly. And it's going to use gravity to pull the things that are more dense to the bottom, then things that are less dense will stay towards the top. So if you see here, right, the least dense component of all this is plasma. And as you can see, it's about 55% of the total whole blood. Now, in the middle, there's this small part here that is what's known as a buffy coat. And this is going to be less than 1% of the total blood. And basically, this buffy coat is made of white blood cells known as leukocytes and essentially platelets, which, like we'll learn more about, are just fragments of cells. And this, as you can see, is just this thin whitish layer between the red blood cells on the bottom and the plasma layer on the top. Now, as you can see in this bottom part, right, this essentially, if you can look in here, is going to be where all the erythrocytes are. And so this is about 45% of the blood. And you can look at that as a hematocrit. And what that is, it's basically the percentage of blood volume that's considered red blood cells. So if you can see on here, They're looking on the average is about 45%. Now that differs a little bit between male and female. Which one do you think is going to be bigger, male or female? Hopefully you realize males are bigger in this sense. They're at 47% versus women at 42%. And both of these are plus or minus about 5%. Why do you think that's the case? Hopefully you figured out that on average men are bigger than women, and that this percent overall that are erythrocytes is based on total amount. Men generally have more muscles, they need more oxygen, they're bigger, so you're going to have more erythrocytes there to help move them around. Now, the physical characteristics of blood and how much volume there is, essentially. Blood is a sticky, opaque fluid, and it has a metallic taste. So if you've ever bitten the inside of your mouth, you will know that it kind of tastes a little metallic-y. The color varies with O2 concentrations. So essentially, if you have high O2 concentrations, this is what's known as O2 loading. It produces what's known as oxyhemoglobin. Oops. And we're going to talk here a second what hemoglobin is. This is basically a protein that you find in the middle of a red blood cell, and it likes to attach to oxygen, thus the oxy part of this. Whenever this happens, you're going to get a color that's considered a scarlet red. Really, really, really bright red. And this is what you see whenever you cut yourself, right? Now, if you have low O2 levels, right, this is going to be the blood that leaves from your tissue going back to your heart to be basically re-inundated with oxygen. This is what's known as O2 unloading, because O2 is leaving, it's not present, right? If you no longer have oxygen, it's called deoxy, and basically it's in the form of hemoglobin. And this is what you're going to see, a dark red. And that's what you really see underneath. Now for me, it might look kind of bluish because it's the color of that blood through layers of skin. So it's going to change kind of what the color looks like. Now the pH of blood is generally somewhere between 7.35 to 7.45. And on average, the blood constitutes about 8% of the body weight. And so for males, they normally have between 5 and 6 liters of blood. You think females are going to be more or less? It's small, slightly smaller, with 4 to 5 liters. Now let's talk really quick about the different parts of blood. So we're going to start here with blood plasma. So blood plasma is a straw-colored sticky fluid, right? This is not the actual cells. It's just what the cells are bathed in. About 90% of it is water, and there are 100 different solutes that are there, right? And so when I mean about solutes here, we're talking about what type of ions or molecules you can find floating around. Now for blood plasma, its overall function here is to suspend cells. That should make perfect sense because we have all those form elements. To transport different molecules. So you might have hormones. You might have cells. Or you might even have things like heat. Remember, we were talking about the function of blood in general. And so now we're going into looking at the individual parts of the blood and what every part actually does in that big scope. So when we're looking at overall molecules, it moves around electrolytes, right? So you have within it things like calcium, potassium, sodium, and chlorine ions in there. You have nutrients that are flowing around to cells. So things like amino acids, simple sugars like glucose or fructose, fats like cholesterol, or even vitamins like vitamin A. You have things like non-protein molecules, substances that are there. So again, amino acids fit into this. You have things like uric acid, urea, or ammonia. These are all different waste molecules that need to be removed. You have things like creatine that is in there, along with creatinine. So these are all different molecules that are important for cell function. Now the last part that you'll get within here, and I'm going to remove all of this. So if you need any more time throughout this, just hit pause and you can fix, or as you go through this, right? If at any point you just need to take a break, the great thing about this is that you can pause and walk off. And so the last thing we have are plasma proteins. And so there's 3 main proteins in here, right? So you have things like albumin. About 60% of the different proteins are albumin. And this guy is important because he's going to help maintain constant blood volume. And he does that by allowing the regulation of osmotic pressure. And this guy is basically produced by the liver. So depending on the concentration of this protein is going to basically give solute concentrations, and osmotic pressure is the idea of water moving across a membrane. So albumin is one protein that helps to keep water levels balanced. Globulins, which account for about 35% of the total proteins, essentially are important in transporting lipids and fat-soluble vitamins and used to help with production of antibodies. So basically, these guys are known as alpha, beta, and gamma globins. So alpha and beta here are important because they transport lipids and fat-soluble vitamins. And they're produced by the liver. And then the gamma globulins are used for antibodies. And they're produced by the lymphatic system. Now, I said there's 3 major proteins, so I'm going to go ahead and get this. And the last protein we're going to talk about is the one that's the largest out of all these, but smallest in number, which is fibrinogen. Really important, and you'll realize once you're doing the case study how important it is. Only 4 to 5% of the total protein count in blood is fibrinogen. and it's made in the liver, and it's important for blood clotting. So this guy essentially will be converted into fibrin for blood clotting later. So those are all the important parts of blood plasma. Now let's talk about the formed elements within the blood plasma. So you can see on here in this beautiful picture on there all the leukocytes, the platelets, and erythrocytes. And so there's 3 types of overall formed elements. Only leukocytes, or white blood cells, are actually complete cells. When you look at it, we're going to and we talked about this in class, red blood cells, they don't have any nucleus or other organelles. Platelets are just cell fragments. So the only one that's complete cells are essentially white blood cells. And you can see the nucleus in all of these because those are the dark stained areas. Most formed elements survive in the bloodstream only a few days, and we're going to talk about how long each one of these lasts. And most blood cells originate in the blood bone marrow and do not divide. It's hard to divide if you don't have a nucleus in some of these. So we're going to talk about this. So let's start with erythrocytes. And so the structural characteristics of erythrocytes. You can see here that they are essentially small in diameter, and they are important for O2 transport, right? They are biconcave, so you can see that they cave in on both sides, right? They are anucleated, which means you don't see a nucleus there at all, right? There are no organelles. What they are filled with is hemoglobin. And hemoglobin is there, if you remember, we were talking about the globulins that are there, and part of it has come together to make hemoglobin. And so hemoglobin is important for actually moving oxygen around in the body. Red blood cell diameters are larger than some capillaries, and so they contain in their membrane this protein called spectrin. And spectrin allows these guys to be very flexible, to be able to move through these really small capillary vessels, right? Now, because they're anucleated, these guys do not perform mitosis, which means they do not divide. Now, these guys offer a huge surface area. Do you see how all of this surface area versus relationship to volume? So that means that they're really good for gas exchange. And basically, none of the oxygen is consumed because there's no mitochondria. So they're really efficient being able to grab the oxygen because they're tiny and they have a large surface area but a very small volume. About 97% of the cell volume is hemoglobin, so it's going to be able to grab onto that oxygen effectively. It's going to be able to get into very small vessels because of the spectrin that's on the outside of that membrane, that protein that allows them to be very, very flexible. And again, because a majority of the cell is hemoglobin and it has such a huge surface area to small volume, the oxygen can escape it very easily and go into the tissue. So erythrocytes are very, very cool in that regard. And again, they have no mitochondria, so they are anaerobic. They're not going to consume the oxygen that they're carrying to make energy. They're really cool when you think about it. So red blood cells, or these erythrocytes, are dedicated to respiratory gas exchange, right? So this right here shows you a hemoglobin molecule. And it basically has 4 different chains, or polypeptide chains. And so if you look on here, here's one right here. Here is another right down here. Here's the third. And here's the fourth. So there are 4 of these polypeptide chains, and polypeptide chains are just protein chains, right? And within each one of these guys, you can see right here, there's one over here, there's one in here, and there's one right there. They have what are known as these heme groups. So the protein is the globulins, and they're alpha and beta, slightly different conformations, and with each one of them they have a heme. And these heme groups essentially are shown right here. And in the middle of them is iron. And that is what will bind to oxygen. And it's reversible. You don't want it to be irreversible, because if it's an irreversible binding, the oxygen is going to bind to the iron and then it won't let it go. And that's what we don't want that to happen, right? We want it to be able to bind and then easily leave later when we need it to. And so essentially that's what's happening here, right? And so this iron, depending on the electrons or the amount of electrons there, that's what is responsible for the color change. Okay. And basically the hemoglobin values for male and female, again, do you think they're going to be exactly the same? No, they're a little different. Think about body size and how much muscle is there. So for basically men, it's 13 to 18 grams per 100 mils or milliliters. And for females, it's 12 to 16 grams per 100 mils. So women on average have a little bit less. Now that we know why erythrocytes are important, let's talk about how we make them. Because we know we don't make them by mitosis, right? So actually talking a little bit about their formation is important. So the formation of all blood cells is a process called hematopoiesis. This occurs in the red bone marrow. That's essentially composed of reticular connective tissue and what's known as blood sinusoids. In adults, this is found in the axial skeleton and the girdle and the proximal epiphysis of the humerus and femur. So it's basically found in all those areas. Last semester that we denoted, that was essentially red bone marrow, that spongy bone that you find red bone marrow in. So the stem cells that all these form elements come from are basically what are known as hemocytoblasts. And these are basically hematopoietic stem cells. And depending on the hormones and the growth factors that are there, they are going to basically specify which pathway that these stem cells are going to go, aka what type of blood cell we will end up producing. Once they've been committed, they can't go back, right? And so basically, and we looked at these a little bit when we were looking at bones in part 1, but new bone cells enter blood sinusoids. And so this is essentially what happens. Now, the areas that we find in adults where we make blood cells is a little different than where we see in embryonic fetal development, right? In the embryo, the red blood cells essentially are made in the yolks, the fetal liver, and the spleen. So this is before birth. Now, once birth happens, then it kicks in to start making red blood cells and the red bone marrow. But there are a few different areas whenever. Now, the blood sinusoids that we're talking about are basically irregular shaped blood vessels. That are normally found in the liver. So now that we've talked generally the process of all blood cells, let's talk about production of just erythrocytes here. So when we're making erythrocytes, it's the process of erythropoiesis. And that's the formation of making red blood cells or erythrocytes. It takes about 15 days, guys. And so you see, basically, we have that cytoblast that we initially have. And whenever we want to start making erythrocyte, this is going to end up transforming into an amyloid stem cell. And again, this is based on different hormones that are there. We're going to talk a little bit about hormonal control once we get through this process. There's a hormone that helps to stimulate this process called erythropoietin, EPO. But essentially all blood cells start from this stem cell here. And then what we got is once we have this myeloid stem cell, it's going to transfer itself into a pro-erythroblast. And at this point, it becomes a committed cell. And you can see it says erythroblast, blast being these birth cells or these beginning cells, right? Now, these guys are going to divide a lot. At this point, you can see that they have a nucleus here, right? So they're going to divide a lot, and essentially they're going to be made into what's called a basophilic erythroblast. And these guys basically synthesize a lot of ribosomes because we want to make a lot of proteins to get ready to essentially form these red blood cells. Now, these guys are very blue in nature because of the ribosomes. As you can tell, as they're going through here, they're starting to change color a little bit. And at this polychromatic erythroblast stage, you'll notice that it's starting to turn a little bit more red. And that's because, essentially, they're starting to produce and accumulate hemoglobin. And so when you go to dye these guys, that red hue and everything else, or it starts showing this red because essentially you have all that iron that's there. Now, as you continue to make these cells and the hemoglobin continues to essentially accumulate, you'll notice that they start becoming extremely pink. See, this is a red hue, right? Now they're becoming this very much this pink color. Because at this point, they contain mostly hemoglobin. And do you notice what's happening here? And these are what are known as orthochromatic erythroblasts. They are starting to essentially eject most organelles. The nucleus is going to degrade, and once that happens, it's going to cause this guy to go into a concave shape. So the shape of it has to do with there's nothing there to kind of push those sides out. Now at this point you get a reticulocyte. They still contain a small amount of ribosomes. And you end up this takes basically about 2 days over here for the ribosomes to degrade, and you become a mature red blood cell. You can see that there. This reticulocyte here, you can use the reticulocyte count to indicate the rate of red blood cell formation. Right? So that's essentially the stages of erythropoiesis, which is a form of the hematopoiesis that we just talked about beforehand. Now, how do we regulate, and what are some of the requirements for red blood cell production? So, if your tissue becomes hypoxic, here, okay, which means there's a reduction in the amount of O2 that's there. So, hypoxia can come from basically decreased red blood cell numbers due to bleeding out, hemorrhages, or some kind of increased destruction. You can have an iron deficiency, so you have less hemoglobin per red blood cell. You could be at high altitudes or have lung problems such as pneumonia, and so that can cause your tissue to go to hypoxia. So any of these things that can cause the reduction of oxygen levels. And so when that happens, this is going to basically release what's known as EPO. EPO is erythropoietin. And this is a hormone that helps to stimulate erythrocyte production. So EPO, or erythropoietin, is a hormone that gets released in times of tissue hypoxia or reduced oxygen levels. Now, if you have too many red blood cells, the tissue can become too viscous, or otherwise known as being too thick. And so here, basically, they would inhibit that hormone, or EPO. So this can happen too many erythrocytes, or it becomes too thick, or if oxygen levels are too high, right? You don't need a lot of EPO, so it slows down. Now, on average, 2 million red blood cells are made per second. It's pretty effective. And basically, EPO can help to increase or help for erythrocytes to mature faster. And so things like not only blood viscosity or hypoxia controls rate of EPO production, but you can go and look at, and we're going to talk about the balance between making red blood cells and destruction as based both on hormonal controls and dietary requirements. For instance, testosterone enhances EPO production. So that is one of the reasons why when we're talking about how much red blood cells and that men have more red blood cells, it's also due to their hormone levels. And so let's talk a little bit and look at erythropoietin mechanisms for regulating erythropoiesis here, right? So for instance here, this is if you have some kind of homeostatic imbalance where the oxygen levels get reduced here, you see? So the stimulus here is a hypoxia. It can be due to decreased red blood cell count, decrease in the amount of hemoglobin, or the availability of O2. And basically from there, the kidneys and liver to a small extent will release EPO. EPO is going to act on the red bone marrow that's located in spongy bone. Basically, that's going to enhance red blood cell formation, and it's going to get more red blood cells carrying O2. So essentially, the stimulus of hypoxia will be ridden, and O2 levels will fall back. Now, once O2 gets high enough, then it'll decrease the production of EPO. And so that's kind of the balancing act that happens. Now, some athletes have been known to abuse EPO. And this is essentially, if you look, Lance Armstrong, he won the Tour de France quite a few times, and they figured out that his drug of choice was EPO. And scientists claim that it doesn't work. Other people claim it does. Basically, the use of EPO increases hematocrit, which is a percentage of red blood cells that are in your body, which allows athletes to increase stamina and performance. There is a dangerous consequence of doing this because if you realize if your average red blood cell count is 45%, and it can increase it up to even 65%, this is going to make your blood even more sluggish. It can cause clotting, strokes, and heart failure. The reason why they believe this is helpful, especially in things like professional cycling, is because if you have more red blood cells, you can get more oxygen to your tissues, reducing the fact that you will end up going into anaerobic respiration or fermentation, making lactic acid. So your muscles are going to be able to perform at a higher level. But it's still kind of being researched as to whether it's extremely effective or not. Now, the dietary requirements for red blood cell formation or erythropoiesis, it's important that we have amino acid, lipids, and carbohydrates. Imperative that you have iron. And so iron can be found in both forms. It can be found basically as ferritin or hemosiderin. And free iron is extremely toxic to the body. So it's stored or it's transported throughout the body, always attached to a protein. If not, it can interact with things and it can be extremely deadly and toxic. So if you're storing for later to basically make red blood cells, you're storing in the form of ferritin or hemosiderin. If you're transporting it throughout in the blood, it's going to be bound to transferrin, so that it can't interact with things. B12 and folic acid are necessary for DNA synthesis. for rapid dividing cells, such as the developing red blood cells. As you know, we don't perform mitosis later. So it's very, very important for us to have those to allow for the initial red blood cells to be made. And so if we're making red blood cells, it's also important that we remove them as well. And so the fate and destruction of erythrocytes, just to let you know, their lifespan is between 100 to 120 days. They are anucleated, as I've stated before, so they can't synthesize new proteins or grow or divide through mitosis. As these red blood cells become older, they become fragile, and essentially the hemoglobin begins to degenerate. So these guys can also get trapped in smaller circulatory channels. They get trapped in the spleen. If they're dying and starting to break down, in the spleen, macrophages can actually engulf them and get rid of them. And what they're going to do is they're going to separate within the red blood cells the heme, the iron, and the globin. So let's talk about how this actually works. So if you see here, this little picture is going through the overall sequence altogether. So if you start here, you get basically low O2 levels in the blood, stimulate the kidneys here to produce EPO. EPO levels rise in the blood, and essentially erythropoietin and necessary raw materials in the blood promote erythropoiesis in the red blood bone marrow. And here's an example where it's going into the epiphysis of this bone. Now they're going to enter the bloodstream here, and they're good for about 120 days, right? As they become aged and damaged, they can be engulfed by macrophages or the spleen and liver, and it's broken down. So the hemoglobin, which is what's important in moving around the oxygen, gets divided into globins and heme groups. If you remember when we talked about essentially globins being in the plasma, so it can go into amino acids and be put right back into the plasma. The heme is going to be broken down. And so the iron from that heme group is stored as ferritin or hemosiderin, right? And for it to be allowed to be reused. So you can see it goes into the liver, and then if it needs to be, iron is bound to transferrin and released to the blood from the liver whenever it's needed for erythropoiesis, right? Now, the heme itself, not the iron, is broken down. So it's taken up, and basically the heme is degraded to this yellow pigment called bilirubin. You can see that is. And the bilirubin gets picked up by the liver here. This bilirubin is then secreted into the intestines through bile. And it's degraded to a pigment that's known as urobilinogen. And then that basically is metabolized to stercobilin. You can see that right there. And that's going to leave the body in the form of the feces. And this is a brown pigment. That's why your feces is brown. So it's all because of the red blood cells, or the heme group within your red blood cells, that allow your feces to be the color that it is, on top of the pigments that you get from food. So this is how essentially red blood cells are broken down. Now let's really quick talk about the different types of disorders that are known for erythrocytes. So hemorrhagic anemia is an anemia that's due to rapid blood loss. This happens due to things like severe wounds, and it's treated by blood replacement. So this, you get stabbed in the jugular, something like that, which is really, really bad, and chances are of you surviving that very, very slim. But this is get stabbed in the stomach, get stabbed somewhere where it's going to essentially allow for rapid blood loss. Chronic hemorrhagic anemia is more of a persistent blood loss. So think of things like hemorrhoids or bleeding ulcers. So basically the primary treatment here is to stop the bleeding. Iron deficiency anemia can be caused by hemorrhagic anemia. So it can be caused by these guys, or it can be caused by low iron uptake or impaired absorption of iron. So the kind of red blood cells that we produce here end up being called microcytes, and they don't function the same way. They're small, they're very pale in color, and they cannot synthesize hemoglobin because they lack the iron. So here, the best way to treat this is iron supplements. So this is one thing that you have to worry about here. That a lot of people that are vegetarians or vegans can suffer from that if they don't make sure that they're getting the correct amount of iron in their diet. Okay, so I'm going to read. Pernicious anemia. This is a autoimmune disease. And this basically is going to destroy the stomach mucosa. That produces intrinsic factor. Intrinsic factor is needed for B12 absorption. And if you remember when we talked about folic acid and B12, it's needed to help red blood cells divide. So basically without B12, red blood cells enlarge but can't divide. So they result in these red blood cells that are known as macrocytes. And so to fix this, you can do treatment is B12 injections. And this is another particular type of anemia that can be a problem for vegetarians. So make sure that you get diets that are well suited for B12 so that you can have your red blood cells become robust and functional. Renal anemia. So this is a lack of EPO. This is all normally due to a renal disease. Remember, EPO gets made by the kidneys. So they can't produce enough EPO. So here is treatment is synthetic EPO. Aplastic anemia is basically due to destruction or inhibition of red bone marrow. It can be caused by like drugs or chemicals, radiation, or viruses. All formed elements are affected through this. So not just erythrocytes, right? So this is one that it's not just a reduction. It causes problems in anemia, but it also causes problems with clotting and immunity, right? Think about it. If you're having problems making all formed elements, that means you're having problems making thrombocytes, which are important for clotting, and leukocytes, which are important for immune functions. So it's going to be a problem all the way around. With this guy, you can help with short-term transfusions, and long-term, you're going to want to do stem cell therapy. Hemolytic anemia. So this is too many red blood cells destroyed. Thinks lysis. So this is when we're destroying red blood cells prematurely. It can happen from an incompatible transfusion or infection. A lot of times, this can happen due to genetic disorders. And this can result in both thalassemia and sickle cell anemia. So let's talk about these two guys really quick. So this is normally found in people with Mediterranean ancestry. And basically, if you remember, one of the globin chains is absent or not working. Here, you're going to notice that basically the red blood cells are very thin, delicate, and deficient in hemoglobin. And so the severity of this is very varied. In severe cases, you can get monthly blood transfusions. Now in here, sickle cell anemia, basically it's a 1 amino acid change. And that's within one of the globin chains. And so basically what happens is it misshapes the red blood cell. It goes from being a circle to looking like a sickle. And so you end up getting reduced O2 transport, and it's very painful. So you see this mostly in Black people in the African Malarian belt and their descendants. So if someone has descendants from that region, there's a really good chance that they could carry this. Now, carrying it means that you only have one disease gene, so you're not going to see it. It's whenever parents come together that have two disease genes that their children can essentially have it. So it's always good to get checked for this. So it's beneficial. People with sickle cell don't contact malaria, and malaria kills about 1 million people each year. So if you're in those regions that have malaria, it's been also seen in certain parts or the water in certain parts of India as well, right? And so if you have this and it can protect you from it, it's very beneficial. The problem is, especially if your descendants have come over here, we don't have a problem with malaria. So this is essentially genetic change that's not advantageous over here for you. And so the treatment for here is basically blood transfusions, and essentially you can inhale nitric oxide to help with this too. And the last one, polycythemia. And so this is abscessed is an abnormal excess of red blood cells. And so this is going to increase viscosity of the blood and cause basically sluggish blood flow. And so in here you may see hemocrats as high as 80%. So you hear a lot of times they'll do what's known as therapeutic phlebotomy. And so most of the time this originates from what's known as polycythemia vera. And this is bone marrow cancer. And this bone marrow cancer leads to high or excess amounts of red blood cell. Secondary polycythemia can result from, example, high altitudes or increased EPO. And secondary is also known or result from blood doping, like we talked about earlier. So these are all the homeostatic imbalances that can basically proceed from or originate from erythrocytes disorder. So we're going to go ahead and pause here, and we will get started with leukocytes next.